Hey, hello bio, this is Mr. B. We're continuing on with section 1 of DNA, and we're looking at part 2. So we're going to look at another experiment that helped us understand what we know today about DNA. So this experiment was done by the people of the name Hershey and Chase. So this isn't our chocolate person that's making this. This is a guy by the name of Alfred Hershey and Martha Chase, and their reason how they kind of came into DNA was that they studied viruses, which are non-living particles, which actually that's kind of up for debate whether or not viruses are living or non-living, which we will get to that later in the year. And they are smaller than a cell, and they can infect living organisms. So I think we've all known viruses, and the flu is caused by a virus and things like that. So we've all at least heard of viruses before. So these people, Hershey and Chase, they studied viruses. And one specific virus that they studied is known as a bacteriophage. And now this is kind of, again, it sounds complicated, but it's really not that bad, because it's simply a virus that infects bacteria. So when you see the word bacteriophage, it's not a bacteria, it's a virus that infects bacteria. And they are composed of DNA or RNA core and a protein coat. So they're pretty simple. They either have DNA or RNA and protein, and that makes up their entire virus. And kind of how these bacteriophages work, when they enter a bacteria or a bacterium, the virus will attach to the surface of the cell, and it injects its genetic information into it. So we're going to see some pictures here in a second. It basically looks like a little spaceship that lands on the cell, and it'll inject its genetic information into it, and therefore infecting our bacteria. And the viral genes produce many new bacteriophages, which eventually destroy the bacterium. So they basically hijack the bacteria to make new viruses for them. And then when the cell eventually will split open, and hundreds of new viruses burst out. So inside, all these new viruses are being made, and once the time eventually comes when there's no room, the cell literally breaks open, and then all those new viruses go out to do the same thing. And Hershey and Chase, they wanted to determine which part of the virus entered and infected the cell, and they would learn whether genes were made of protein or DNA. So if they could determine which part of the virus entered an infected cell, they would learn whether genes were made of protein or DNA. Because up to this point, we didn't know what the composition of our genes were. So in order to do this, they grew viruses in cultures containing radioactive isotopes of phosphorus and sulfur. And the purpose of phosphorus and sulfur is that one will either just simply bind to the protein and one will bind to the DNA. So it's basically just like a big way for us to mark our different parts of that bacteriophage. So I think, okay, yes, and sulfur is going to connect to the protein. So if sulfur was found in the bacteria, it would mean that the virus's protein had been injected into the bacteria. So you can see all those little pink dots, that's all the sulfur connected to the protein that's connected to the protein of the bacteria. And the sulfur is connected to the DNA. You can see, again, it looked like a spaceship there on the left, and you can see all the pink dots are now on our DNA, which is on the inside, and therefore it is marked, and that would mean there would be high amounts of phosphorus if our bacteria was infected with DNA. And so their results were nearly all the radioactivity in the bacteria was from phosphorus, and therefore Hershey and Chase concluded that the genetic makeup of the bacteriophage was DNA, not protein. So after you can see here in our two experiments, no radioactivity when the ones that were treated with sulfur, lots of radioactivity with the ones that were treated with phosphorus. Therefore, we know that the genetic material of our bacteriophage was DNA and not protein. So that's it for this video. Let me know if you have any questions.